Hello, my name is Aisosa Omere, and I'm in Dr. Buonomo's lab here at UTA. And today I will be presenting my project, diazo compounds and pH control, separating side chain reactivity and the C-terminus. Of the 20 canonical amino acids, only aspartic acid and glutamic acid bear a carboxyl terminating side chain. As a result of the peptide bond, proteins also contain a carboxyl terminus, otherwise known as the C-terminus, and amino terminus, or the N-terminus. The physical properties of carboxyl functional groups make them a highly versatile functional group capable of engaging in molecular interactions with a wide variety of complementary functional groups. Aspartic acid and glutamic acid and the C-terminus exist as their respective ionized conjugate bases at physiological pH 7.4. This equilibria has limited the number of chemical probes that selectively react with these functional groups. But here, we demonstrate the ability to separate the side chain and C-terminal reactivity by harnessing the chemoselectivity of diazo compounds and pH control. Here we see with the aspartic acid side chain a pKa that can vary from 3.0 to 4.2. And with the C-terminal pKa, we see a range of 1.7 to 2.4 as a result of the electrostatic interactions within the peptide. Using a chemoselective probe like diphenyldiazomethane depicted here, we can chemoselectively react with carboxyl functional groups. And on top of that, by utilizing pH control, we can select which carboxyl group we can interact with within the peptide sample. So we did a screening experiment with 1 millimolar of peptide and 10 millimolars of DP-DAM, and we tested from a range of 2.8 to 8.0 in 50-50 acetonitrile and buffered water. And we did this reaction for 4 hours at room temperature to see whether or not we would get a labeling. At pH 4.0, we had a side chain specific labeling that was confirmed by tandem mass spectrometry. And here we have the Benz hydromodification, so the addition of diphenyldiazomethane, depicted by the Y9 and Y10 ions in the tandem MS diagram here. And so then again we did the experiment and we saw labeling at 2.0 that was C-terminal specific. For the selectivity of this diazo compound, it's driven by the mechanism in which the reactive resonance form, the carbene, can abstract a proton from the carboxylic acid. This forms a carboxylate that can then act as a nucleophile and displace the diazo functional group as molecular nitrogen to form our ester. The carboxylic acids are able to react because they are able to participate in this key mechanistic step in which the carbene resonance form is able to deprotonate the carboxylic acid. Carboxylates are deprotonated and therefore are unable to react. Then, utilizing our pH control, we can enable C-terminal specific reactions. And so here at pH 4.7, reacted DP-DAM with RGDC peptide to get a protected aspartate, and we have a free C-terminus. Then, undergoing an EDC coupling with propargylamine, we add the propargyl group, or the alkyne here, which can undergo our subsequent utilizations. Utilizing a copper catalyzed azide alkyne cycloaddition, we immobilized RGDC peptide that had been alkynylated onto an azide bead, or a bead whose surface was functionalized with an azide. This reaction was then QC'd with a fluorophore that was then the results were then confirmed with epifluorescent microscopy. Our next step is to develop a C-terminal selective probe utilizing the same structure as DP-DAM, or diphenyl